Effect of Substrate Concentration on Rate of Reaction by Jonathan Wong, Arashpreet Gill, Alyssa Peterson, and Alyssa Garrido. In the decomposition reaction of hydrogen peroxide into the products of water and oxygen gas, the enzyme, catalase, drawn and labeled in red, can catalyze the reaction, meaning that it can speed up the rate of reaction by lowering the activation energy. When the substrate, or the reactant that the enzyme acts on, labeled in blue, comes in contact with the enzyme, it will enter the active site, where the magic happens. There are also other factors that affect rate of reaction, including temperature and pH. With temperature, there is an optimal temperature in which the substrate will come in contact with the active sites of the enzyme at maximum efficiency. With pH, there is also an optimal pH in which the hydrogen ions will not affect the enzyme itself. If one of these factors is too high, the enzyme will denature.
Research question: how does the substrate concentration affect the rate of reaction? Hypotheses: if the concentration of hydrogen peroxide increases, then the rate of reaction will increase. If the concentration of hydrogen peroxide decreases, then the rate of reactions will decrease.
Part 1. Making beef liver solution. Cut and measure 0.25 grams of beef liver and 25 milliliters of distilled water. Add the beef liver and 10 milliliters of the distilled water into the mortar. Using a pestle, grind the beef liver until there are no more chunks of beef liver remaining. Add the 15 milliliters of distilled water to the mortar. Part 2. Making hydrogen peroxide solutions. There are 2% solutions that need to be made, 1% and 2%. To make the 1% solution, 12 milliliters of 3% hydrogen peroxide are needed, and for the 2%, 24 milliliters are needed. For the 1% solution, 24 milliliters of distilled water are added, and for the 2%, 12 milliliters of distilled water. The total volumes for both should equal 36 milliliters. The 3% hydrogen peroxide solution is the stock solution. Part 3. Conducting the reaction. 5 milliliters of the hydrogen peroxide are added to a test tube. Using a transfer pipette, 5 milliliters of the beef liver are added to the test tube. The height was collected after 15 seconds, or if it overflowed, the time it took to overflow was measured.
This is a table of our collected data. When the 1% solution reacted, the bubbles never went over the top, and the height shown is recorded after 15 seconds. The average height of the bubbles was 71 millimeters. When the 2% and 3% solutions were reacted, the bubbles did go over the top, and the time shown in the table was the time it took to go over the top in seconds. The average time was 1.91 seconds and 0.94 seconds. When the rates of reaction are calculated, the 1% rate of reaction is 4.7 millimeters per second. The 2% is 50.3 millimeters per second, and the 3% is 102.1 millimeters per second. When the rates of reaction are graphed, as shown here, the graph shows an upward trend. Our experiment's results show that our hypotheses were supported. The first hypothesis is that the rate of reaction increases as the hydrogen peroxide concentration increases. As the hydrogen peroxide concentration goes up from 1% to 2%, the rate of reaction increases from 4.7 millimeters per second to 50.3 millimeters per second. This shows an upward trend, which is further solidified by the graph. The second hypothesis is that the rate of reaction will decrease as the concentration of hydrogen peroxide decreases. This is shown in the graph and in the calculated rates of reaction. As the hydrogen peroxide decreases from 3% to 2%, the rate of reaction decreases from 102.1 millimeters per second to 50.3 millimeters per second. There are many weaknesses in this experiment.
The first weakness is approximating the weight of the liver on the scale because it was hard to cut.
To improve, we could use a sharper tool and a more accurate scale, and also a more blunt pestle.
The second weakness is that the liver left residue when being transferred from the weigh boat into the mortar, which changed the concentration of the solution.
To improve this, we would rinse the weigh boat with part of the 25 milliliters of distilled water.
Third weakness is that only the top of the hydrogen peroxide solution reacted in the narrow test tube.
An improvement would be to reduce the amounts that needed to be combined, or use a larger or wider test tube.
The fourth weakness is that the water made it difficult to see the chunks of beef liver. As a result, there were still some chunks remaining at the end.
To improve, we would smash the liver before inserting the water.
The fifth weakness is that the phone only went to 2 places after the decimal and had a lag time.
An improvement would be to use a stopwatch or a timer with more decimal places.
The sixth weakness is that the liver has volume, making the solution greater than 25 milliliters.
To improve, we would account for the volume the liver takes up and just fill to the line rather than having a preset amount.
The seventh weakness is that 15 seconds may have been too long and the reaction may have leveled off earlier.
An improvement would be to record the height at more intervals.
The final weakness is that the 106 millimeters in our data was extraneous.
To improve, we would account for the liquid that turns into bubbles and water.
In the future, there are many experiments we could do to expand our knowledge of catalase. One such experiment is to compare the rates of reaction of the different types of liver, since we had only tested beef liver. Another experiment is to compare the rates of reactions of plants to animals.